Great, thank you very much, and thank you for the organisers for the invitation to speak. So, to briefly introduce myself, so I am an immunologist by background and a patent attorney specialising in biologics and particular cell and gene therapies. I'm a fractional head of IP at Cartesian Therapeutics, which is a late-stage CAR-T mRNA cell therapy company, but also patent attorney at Evolve Intellectual Property. So I'm going to, for the panel discussion on mRNA, introduce the concept, why it is such an exciting technology, what the actual scientific and commercial landscape currently is, and why things are changing. And I think from my background, so I did spend 6 years at AstraZeneca in-house, and there I learned not only how pharma companies think about IP strategy, but how that has to be linked in with all of the scientific and commercial goals as well. If the first question that investors or pharma companies ask your biotech is, does the science work? The next question is always, what IP do you have to protect that science? And thinking about that in terms of the mRNA technologies, this is a very, very complex scientific area, but also very complex from a commercial point of view, and therefore also from an IP point of view. So without further ado, first thing, RNA therapeutics are not new. We have history going back. The first ones in terms of RNA were the antisense oligonucleotides that were 2016 approved. Then that sort of turned into the siRNAs, and these therapeutics are modifying gene expression, essentially. So you want to knock down a gene that's harmful, you put in an siRNA to decrease expression. mRNA really came on the picture with the COVID vaccines that we all know, hugely successful, very, very effective, multiple billion-dollar products. And out of that is where we've now got the new interest in the mRNA therapeutics, doing something different than just expressing viral proteins as in the vaccines, but actually expressing therapeutic molecules instead. And there are challenges with a move to that more therapeutic protein expression, in that for the vaccines, generally they're packaged in LNPs. Doesn't hugely matter in terms of targeting where those LNPs go, but for therapeutic proteins, a big challenge is going to be targeting your LNPs, and that is where a lot of the center of innovation actually is. And that brings me, so we've already had a great introduction to CAR-T. It's one of my specialities and my interests. So here's a slide summarizing what CAR-T actually does, and you can see why we're going to discuss CAR-T, because this is where mRNA now is making an appearance. But traditional CAR-T is generally DNA vectors that express a chimeric antigen receptor, a targeting molecule within the cell. So you modify the cell to express this targeting protein. Then what the cell can do is go off and find either harmful cancerous cells based on that targeting molecule, or autoreactive cells in autoimmune disease. Then the cell is able to mount a response against that harmful cell. So in the case of cancer, killing the cancer cell, autoimmunity, killing the autoreactive B cells, for example. But what is really key, and why cell therapy is such an effective thing, is that you've got a cell here. You haven't just got like one molecule that's doing a very specific thing. You've got a cell that's able to orchestrate the entire immune system against what it's targeting. And that brings in the power of the patient's own immune system to do effective management of the disease. And this is, as we've seen in the previous talk, why these therapies are able to actually have hugely high efficacy with regards to cancer and an autoimmune disease. But it's complicated. It's very hard therapy to actually administer to patients and to give to patients, and this is because you need to actually extract the patient's own cells to do the therapy. So here the diagram just illustrates how you actually make the CAR-T in the traditional sense. So you take out the patient's cells, you then have to modify them, you have to grow them up, you have to quality check them, and then put them back into the patient. And this takes weeks. The patient has to be in hospital. There's lots of invasive procedures that have to go on. So it's a hard therapy to actually deliver, and it's a hard therapy to deliver at scale as well, because you've got these individual sort of patient journey that you have to go through. In terms of like the challenges in the field as well, it's not easy to work out what you're going to be targeting, how you're going to expand your cells, how you're going to make sure that these are safe therapies. So again, it's a challenging, complicated scientific endeavor. But then this brings us to in vivo CAR T. So this is where all the excitement currently is, because we've got this therapy with normal cell therapy, which has hugely high efficacy, but it has all the associated challenges. In vivo, the idea is, instead of modifying the patient's cells outside of the body, you create a particle, an LNP with an mRNA in it, that you just administer that to the patient, and it will modify the patient's cells in situ, inside the body, so in vivo. And why this is beneficial is that you can make loads and loads of LNPs with mRNAs in them that you can store, keep, administer to lots of different people. You've got scale now. You've got the ability to make a product that can be delivered at scale. But of course, there are lots of challenges as well, scientifically. First, you've got all the challenges from normal cell therapy, what targeting molecules you're going to use, what your payloads are going to be. But then additionally, you've got now the challenges with the delivery vehicle as well. So for LNPs, what is your LNP formulation? How are you going to target those LNPs? How are you going to manufacture those LNPs as well? So it's not a silver bullet. There's a huge amount of work to be done actually taking these products to market as well. But huge excitement. And this slide sort of summarizes some of the big acquisition deals that have happened in the in vivo cell therapy space in the last few years. And what is happening here, and it's very interesting, is that in sort of traditional pharma land, pharma companies might go and license in a product that has some initial clinical proof of concept, license that in, develop that, take that to market. What we're seeing in the in vivo CAR T space is they're buying platforms. They're looking for a biotech company that may not even actually have any clinical data yet, or very limited clinical data, and buying up that platform for a lot of money. And that is a shift, because then the pharma companies has to then actually develop the products and take them to market. But because it's such a competitive field, because there's a lot of potential here, there is, yes, this focus on platform. There is two types of in vivo that I put up here. So we're in an mRNA session, so we're talking about mRNA, but there's a rival approach, which is the lentivectors, which is DNA. And we don't know yet which of these is going to win, whether there's place for both in the in vivo space. Maybe you want one for autoimmune, maybe one for cancer. But yes, so there's currently companies buying lentivector platforms and companies buying the LNP mRNA platform. If you're Eli Lilly, you buy both as well, just to hedge your bets. So where does this all leave us with regards to commercial strategy and IP strategy? It means you have to be thinking very carefully when you're taking your biotech and working out what you're doing with regards to, are you a platform company? Are you a product company? Are you a combination of the two? And how do you actually build out a commercial story that is then attractive to pharma companies who are now maybe looking for a platform that is distinguished from everyone else's and gives you something different, gives you something that is able to actually provide the product that you need. And so what I'm going to do now, quickly, in the last section, is show you the IP portfolios for different types of companies and what kind of strategy they've used to that is linked to whether it's, say, product, platform, or a combination. So this is a chart of a patent portfolio. We call them ammonite charts. So each wedge on this chart is a patent family. Patents last 20 years, so as you go clockwise around the chart, the expiry dates and the filing dates go further and further out. So you can see where the patents are filed and how long they last, what dates they're going to expire. The different colours are the different types of case. So whether your patent covers a product, a composition of matter, whether it covers method of treatment, or treatment protocols, or manufacturing. So this is the patent portfolio for Yescarta, which is an on-market CD19 targeted CAR T cell therapy, so traditional CAR T. And you can see what we've got here is in the gold, the product IP, some foundational IP protecting the CAR, that's the targeting molecule. Then you've got treatment protocol, method of treatment, manufacturing IP that's building layers of protection around this product. So this is the very traditional approach about how you go about protecting your pipeline product. However, so this is the gross yearly sales last year for Yescarta, 1.5 billion. Interestingly, down 5%. It's down 5%, despite all its IP, which has not expired yet, despite all the protections they have. It's down because of competition now from other modalities too. T-cell engagers, different cell therapies coming in. So this is very interesting with regards to what is the value of this IP when we're getting now actually decrease in revenue despite the fact you've got all of it still in force. So this is the IP portfolio for Orna. This is one of the companies that was acquired by Eli Lilly. And this is very, very interesting because here, again, you've got the different types of IP, but you can see they're all very close together. And what you've got is different types of platform IP, protecting different aspects of their in vivo cell therapy platform. So you've got your RNA payload. So the thing about Orna, they have circular RNA. You have the CARs that they've developed themselves to target the cell when it gets in the body. You've got their LNP formulations, you've got their ionizable lipids, all forming part of a platform that can be developed and is very, very attractive to companies like Eli Lilly. And you can see, as they very filed close together, they're filing a lot at one time to really, really build out and protect their R&D efforts. So that was the acquisition deal. And then finally, this is an interesting example. So within the mRNA LNP space, finished, yeah, last slide. So within the LNP space, companies are also protecting just individual platforms for part of the component. So this is the ionizable lipid platform for Acuitas, so part of the big LNP vaccine litigation. And you can see what they've done is very much focus on one type of modality, particularly the ionizable lipids, and they're licensing that out to everybody else. So instead of being acquired, their strategy is have all these different ionizable lipids. We can license these out non-exclusively to lots of different companies as an alternative strategy. So that's just a summary again of thinking where you put your IP within that structure. But the message is that you need to think about not just the really complexity of the science, but also how this interlinks with commercial strategy and then the IP strategy as well. Happy to take any questions.